Hi, I'm Jason Hooper from FixYourGut.com, and today I'm going to be doing part one of my dopamine detox series. So let me just explain what this whole series is going to be about, and in each additional video I'll probably do a recap of this. One of the things that I believe is that we've adjusted our environment so much over the course of the last 100 years that we're under higher levels of stimulus than we ever were throughout our natural history where our species evolved. And I think that having chronic exposure to very strong stimuli, we sort of develop a tolerance over a time, and or it can affect some of our behaviors. So throughout this 30 days, I developed a dopamine detox protocol, and I went on it myself just to test it out because a lot of the clients that I work with, and I feel like some of their behaviors are hijacked because of their brains being subjected to high amounts of dopamine. So why does our brain make dopamine in the first place, and how does that affect how our bodies respond to the environment? Dopamine is part of the reward system in the brain. It activates when something occurs that is going to help us to survive. So today we're going to be focusing on food and how dopamine affects the sensory specific taste receptors, and we're also going to be talking about the cephalic phase responses. And we'll go, don't worry if you don't know anything about those terms, we'll go into more detail later, but basically how the food we eat affects the reward centers of our brain. And the studies of this on this area are just sort of emerging right now. It's not the best science, but I'm going to talk to you about my experience with it and sort of what I've done with it and why. So of course, throughout human history, we, and not just us, but all animals have a taste response. Even earthworms can detect taste. It's a very important part of nutrient selection, is how something tastes, because basically what happens is the taste buds tell our brain sort of what is making up the composition of this food that we're eating. And so we have the five tastes. We have salty, sour, bitter, umami, and then sweet. And as far as, we're going to go over each of them and show sort of the cephalic phase response for each of the tastes, but we're going to focus specifically for this on sweet, because there was an evolutionary advantage over time to having foods that were sweet in our diet. Foods that are sweet are typically not poisonous, and so you could eat a berry that tasted sweet that was likely not to kill you. And it also, with being able to detect different saccharides on our tongues, we know that it's going to be a good source of calories. Naturally, in nature, there's not a lot of food products that have very concentrated amount of sugars. The natural versions of some of the foods that we eat, or that sometimes they call them the heritage versions or whatever, but that haven't been cultivated and modified over the years, are much less sweet than they are today. And so I think today, with us eating a lot of different sweeteners of all different types, we've desensitized our taste buds and our reward system to what we were initially expected to receive through our environment. Okay, so how does this work? So you're a caveman, you're running around in nature with your club, and you find some berries, and you say, oh, these berries, you may sniff them, and you inspect them or whatever, and then you taste a little bit. Ooh, that's sweet. Okay, and then your brain gives a burst of dopamine because it wants to reward that behavior that you just developed. Say, hey, there's berries here, and you're going to remember where they were, and you're going to know how to find them and how to pick them and all that kind of stuff, and it's going to start a chain reaction to myelinate those memories and sort of solidify that behavior pattern so you can get back to those berries and eat them, because there's a food source there that could be even renewable. And maybe years later, you could be walking by some plants and all of a sudden you're like, oh, there was berries here at one point and they tasted really sweet and I ate them and they were great. But think about all the foods that we eat now that are way sweeter than wild berries and what that's doing to the wiring of our brains. Okay, so let's talk about each taste and talk about the cephalic phase response, which is basically like when your taste buds detect something, you have a reaction in your body. It changes the genes in your body. Some genes flip on and some genes flip off, and we're going to talk about what happens here. So first, I want to talk about salty. Okay, salty. Um, we have to be able to detect salt because we need salt to survive. It's one of the things that affects our hydration. And if you go back to season 1, episode 4, you can discover the hydration video that I did, and I talk about this in more detail. But for now, we're going to talk about more of the sensory-specific taste response and less of the hydration stuff. So basically, this is going to work. On the Ang2 pathway, which sort of detects our level of dehydration in the body. It doesn't detect hydration, it detects dehydration. So when we're dehydrated, we need more water and more salt. This is controlled by the small centers in the hindbrain, so that would tend to indicate too that this gene sequence has been in the genome for millions of years, not just within the course of our species, but many species across the board have this response too, to be able to source salt and be hydrated. So when the body is low in sodium, it activates this gene, flips it on, and then when we're hydrated, it just shuts it down. Okay, so when we consume foods that are salty, it turns off this gene through the sensory input. Okay, and then once that starts to deplete, then it kicks back on this gene, which makes us crave more salt. It's not actually getting the salt in that shuts down the gene, but just even the taste of salt. So if you taste it and then spit it out, it will turn this off until your body realizes it's still dehydrated and then flip it back on. And you'll see some competitive athletes, combat athletes especially, that have to cut weight. They'll use tricks like this with salty and with sour in order to activate a salivary response and to be able to turn off their body's natural dehydration. Now, to help them shed water weight so they can make weight for their sport. Now, this is very dangerous, and we shouldn't be doing this. But, you know, if you got to cut weight quickly, they take these risks, you know, because they think the reward pays off in the end. I'm not sure that it does. So anyway, this gene only detects dehydration, not hydration. That's important to understand because your body only understands when it's dehydrated, not when it's hydrated. There's no rate limit for hydration. So if you're overhydrated, you can have health problems from it, and your body does a very poor job of detecting how that happens. So people that are sipping on water all day, it's not good for you. Drink when you're thirsty, you know, again, and watch my video on hydration. It explains this more. Anyway, there's a couple of other things with salty that kind of is sensory specific with the cephalic phase response. And one of it is the upregulation of leptin, which is a hormone that makes you hungry. And so if you eat salty foods, you tend to eat more. And there are a fair amount of studies that back this up. And this is pretty solid in the literature that salty foods tend to make you eat more until this gene shuts off, and then you're not craving salty foods anymore. So if you're looking to lose weight, maybe don't season your your food quite so much and get your sodium intake in another way. Also, the other thing I wanted to point out about salt is that with most of these tastes, if we exceed a certain threshold, we have a very adverse reaction to it. So if our sodium content is greater than 1.6%, it's going to burn the inside of your mouth. Okay? It's going to burn your taste buds because basically what's happening is you're dehydrating all those cells on the surface of your tongue and mouth, and anything higher than this concentration you're just going to want to spit out. You're not going to want to swallow. It's just way too salty in most cases, unless you're extremely dehydrated, and then maybe you might have a different response to it. But the idea is that if you keep salting your food and salting your food and salting your food, when you reach this threshold, that's kind of the upper limit. And with sweet, we'll talk about later, that's not the case. There seems to be no upper limit for that. And that, again, it affects the dopamine system in the brain, because if there's no upper limit, you need more and more and more of it in order to get the same effect. Okay? All right, now let's talk about sour. Now, sour is an important taste because we can detect the pH levels of our food. Food that's more acidic or has a lower pH tastes more sour. And then with the bitter flavors, foods that are bitter tend to be more basic, and they tend to taste bitter. So basic foods, bitter; sour foods are acidic. So the hormone cascade that, or I guess the cephalic phase response that occurs with the sensory specific taste to sour is otopetrin 1, and this activates when we eat sour foods. Now, why would we want to be able to detect sour foods in nature? It's theorized that this taste evolved because of the way fruit ripen. When a food's not quite ripe, it tends to be more sour, and then because it's converting all the glucose into citric acid, or malic acid, or something in the Krebs cycle that way. If it's holding more of the glucose or sucrose or whatever form the sugar's in, it's not converting quite as much, so that the ratio from sweet to sour is going to change in favor of sweet as the fruit ripens. So perhaps this has been evolved to, if you eat a piece of fruit and we're like, oh, that's not quite ripe yet, I'll come back to this place later. Also with spoilage through the fermentation process, microbes will often lower the pH of whatever food they're consuming because when other spores land, or whenever other bacteria, whatever, reach that particular food, the lower pH helps protect their food from other species so that they can remain dominant. So they find a foodstuff, they build biofilms to protect themselves from the pH, then they lower the pH to try to kill other things off. And that's why a lot of the fermented foods can taste sour. And again, we have an inverse relationship with sour and sweet. So if we eat something very sour, and then we eat something very sweet afterwards, it tastes more sweet. If we eat something sweet and then eat something sour, it's going to taste more sour. So that we sort of have that inverse relationship between the two. There's something called palate fatigue, and I'll talk about this a little bit later in this video, but if you only eat sour foods, you get tired of it really quickly. And even with sweet foods, if you only eat sweet, if you just, it's like, well, it must be great to just eat bowls and bowls of ice cream. But if you look at these people who are do these like competitive eating competitions and that kind of stuff, a lot of times if they're eating something that's really sweet, like ice cream sundae challenge or something, they'll pull out some Sour Patch Kids and they'll pop in a few of them because it's like, I just can't eat anymore. And then, they'll have a little bit of sour, and then all of a sudden they can eat tons more sweet. But that is the cephalic phase response to sour. And again, it's sort of an adverse reaction. If the pH gets too low, we have an aversion to the food. We don't crave more of it. There has to be a balance there, and there is going to be an upper limit too, because if the pH is too low, it's going to burn the inside of your mouth. It's going to burn your esophagus, and so it's not like we can keep increasing or decrease the pH or increase the level of sour for our foods. There's an upper limit for that where we just won't be able to tolerate it. All right, next is bitter. And bitter is a very interesting one. It's interesting from a couple of different points of view because it has a connection to the olfactory system. So like your sense of smell and your sense of taste with bitter, with a lot of these bitter flavors are the exact same things. Bitter is probably the first taste that evolved out of the five, from what we have figured out. And you can actually taste bitter in your sinus cavity. So if you smell like some oregano or some bitter herbs or something like that, you get that taste in your sinus cavity as if it's the same taste in your mouth. Okay, so when we have the fragrant herbs, you use them in a culinary sense. They call them aromatics, right? And it's they're subtle flavors, but you get that smell that it precedes the taste. And most great chefs, they'll add their aromatics in at the very final part of the dish, so that their guests will get that experience, that fragrance experience, the strongest when it comes in. So being able to smell and taste is important with bitter. With bitter, what this does is we have these CALB2, which these are calcium binding proteins, and so when we eat something that's bitter, a couple of things happen. And I think it's because a lot of the vegetables, the leafy greens and stuff that we eat are bitter, and so we anticipate the digestion of these foods. So it increases stomach bile. In fact, that's a hack. If you have low stomach bile, you can eat some digestive bitters, and it makes your stomach produce more acid so that you can digest your food better. A lot of people prefer that over the bentadine HCl supplements or pepsin or something like that, because if they have LERD or GERD or some kind of reflux disorder, it does less damage to their esophagus if they're producing it naturally. So this binding protein, a lot of the calcium that we get also comes from the vegetables that we eat. Like they tend to be higher in calcium than even milk. So it gets our body ready to get this calcium and absorb it, and being able to absorb the nutrients that we're getting from our food better, because it takes a lot more to digest these fibrous plants than it would be for honey or something like that. So yeah, bitters are a really cool one. There also seems to be an upper limit if you have ever gotten a shot of oregano oil or Thymol or something. It's very strong and it can burn you, and so you're not going to just start. Very few people just start like craving bitter things. So the cephalic phase response to bitter seems to be more of a functional thing, where it's like, okay, I ate this, now I got to be able to digest it, not like, oh, I'm craving spinach or kale or something like that. All right, next. Umami it's a similar, and I don't want to get into too much into umami in this video because this is a whole other ball of wax. I just want to talk about I just want to point out the NMDA upregulation, okay, and the term excitotoxin gets used a lot because it activates calcium channels all over the place, and I'm not going to get into the details later, but you know, the umami, most of the umami flavors that we get are through monosodium glutamates, which are either they occur naturally in many different foods. So if you're whether you're eating soy sauce or whether you're using packaged MSG in your food, it's the exact same monosodium glutamate. It doesn't matter where it came from, it does the exact same thing in the body. And depending on the way your brain is set up, you might end up having problems with this, but again, we're not going to go into umami very much. The focus of this video is going to be on sweet, because sweet is something that affects the dopamine center of the brain, unlike all of the other tastes that I just talked about. And I think in modern society, we've artificially increased the sweetness of lots of our foods. There's artificial sweeteners and a whole bunch of other things that come along with it. So the science isn't 100% settled, but I'm going to show you some of the studies that I've got to support my claims about this and why this would be important if you're trying to detoxify dopamine, try to reset that dopamine level, and how this has worked. I tried it myself and it worked for me. You might want to try it too, and there's some precautions to take, so make sure to watch this whole part of the video before you just try this, because you could run into trouble and maybe even damage your health. Okay, so when we get this sensory specific taste, or the SST, of sweet, there are studies that show that it increases the dopamine in our brain 130%. And on top of that, if we want to reinforce those neural patterns that's created by this, which is going to spark myelination of the axons, it also upregulates. So this is for dopamine, and it also upregulates acetylcholine by 133%. So we're getting this upregulation of acetylcholine, which is also reinforcing that, that permanence of the neural pathway that's we use the anthropomorphic term like rewiring the brain, but it's kind of like that. It's like we're putting insulation around these wires that go from point A to point B to reinforce some of the behaviors that we just had. So this is pretty significant just by eating sucrose. We've seen this relationship that's been fairly common here. All right, so the study that this comes from indicates that when the sucrose intake increased from 37 to 112 milliliters per day, which is from 13 to 20 milliliters in the first hour of access, and repeatedly increased extracellular dopamine to 130% of baseline as measured in the NAC shell, that's the exterior plasma that was being calibrated by microdialysis during the first hour of sucrose access on day 1, day 2, and day 21. Okay, so this increase was seen every single day for 3 weeks, according to the study. Okay, and the second part of the study indicates that in the daily intermittent sucrose group, the highest acetylcholine levels, 133%, okay, occurred during the first sample after the sucrose meal ended. So there is some science to support this, and again, it's in animal models, it's not in human models, but it would seem to apply, anecdotally to a lot of cases. I just think it needs to be studied a lot more. So the next study that I want to talk about sort of talks more about the cephalic phase response of sweets and how this doesn't just apply to sugar, but potentially anything that tastes sweet. And this study shows that, well, basically what they did is they took sucrose to represent sweet, sodium chloride to represent salty, hydrochloric acid to represent sour, quinine to represent bitter, or monosodium glutamate to represent umami. And I also just want to point out that whenever subjects are given these 5 categories of foods, they have a distinct preferential treatment for the sucrose over than the other. It's like it's hard to get them to eat all 5 of them because they just want to go for the sucrose. But I mean, think of it for you. If you were offered these 5 things just on a table, just like pure MSG, pure salt, pure hydrochloric acid, pure, well, I guess they were diluted probably they were diluted in a solution. But you know what I mean, though. It's like out of all of these things, which glass of water you want to drink? One with sugar in it, one with salt in it, one with hydrochloric acid in it, one with quinine in it. Like it just doesn't. Those other ones just aren't going to taste as good. So all the animals prefer the sucrose. So let's see what happened. So of course, the organisms elicit an insulin response through CPIR. That's the cephalic phase insulin response. Okay, non-sweet did not elicit CPIR, although rats showed a strong preference for starch, which is a source of glucose. In addition, we studied whether CPIR was related to taste receptor cell activity. So there are enzymes in the saliva that can break down the complex starches into glucose, and you can get a little bit of taste from it. And by doing this protocol, I could actually detect that on my tongue, and I'll talk about that in a second. But it doesn't have the same significant insulin response as pure sugar does. So if you've been fed pure sugar and then you go back to a starch that's not as sweet, you're not going to have the same phase response that you did from sugar. Okay, so the other thing that eating sweet stuffs do is up-regulates insulin. You could think about this kind of as a Pavlovian thing too. Pavlov's dog, they rang the bell and it would increase saliva because the dogs were expecting food. Well, your body does the same thing when you eat something sweet. Your body expects sugar, and so it starts producing more insulin. And you can detect this by measuring your blood sugar before and after consuming an artificial sweetener as well. Get one of those blood glucose monitors, prick yourself, take a baseline measurement, and then drink a Diet Coke with some aspartame in it, and see what happens. Now, if you've generated an insulin response, what does that mean? Does it mean it's going to increase your blood sugar? No, your body is producing insulin, so it's going to be taking sugar out of the blood and into the cells for storage. And so what happens is you drink something that tastes sweet but doesn't actually have any sugar in it to store. It's going to take the available sugar out of the blood and store it in your cells. So you should actually see a drop in blood sugar from drinking a Diet Coke. Now, what happens when your blood sugar drops over a period of time? Well, your body's going to signal, hey, we need to eat more. We need to get our blood sugar back up, and you're going to start craving stuff. But craving what exactly? Craving more sweets, because you need to get your blood sugar higher because you dip below your homeostatic levels. So, let's say you just drink another Diet Coke, and then this is just going to be a vicious cycle of doing this all over the place. So with the protocol that I've done, for 30 days, I eliminated all sweets from my diet, whether they be artificial or natural or whatever you want to call them. Anything that tastes sweet, I wasn't allowed to consume for 30 days. All right, the other thing that I want to clear up right off the bat is that sweets, sucrose, sugars, all that kind of stuff, these are not excitotoxins, and some of the health sites get these confused. It's not an excitotoxin at the same way as MSG is. Yes, it does increase neurotransmitters in the brain. Yes, that's not good, but it's not the same thing. So please stop calling sweets excitotoxins because they're not. Okay, so sugar is not an excitotoxin. Sorry, it's not. Um, Now, that doesn't mean that you get a free ride with sugar or that sugar's any less good or any less bad. It's just don't conflate those terms because they mean different things. Okay, so what happened when I began to eliminate all sweets from my diet? So the goal was that over the course of time, I had been increasing the sweetness of my food, and I needed more and more and more of it to get the same taste as before. And if you use artificial sweeteners, this is a lot easier to do because you're consuming substances like if you consume stevia or monk fruit extract or aspartame, which is an excitotoxin, but we're not going to go down that road. Sucralose, any of those things, they tend to be sweeter than sugar, which is why if you buy it in a powdered form, they tend to puff it up with maltodextrin in order to make it to where a cup, your volume measurements are going to be equal to sugar for baking and whatever, so you can measure it out easier. So you're consuming things that are far sweeter than what you would find in nature, and that's going to continue to increase decrease your sensitivity to the taste of things that are sweet. Okay, so within just a couple of days, I began to notice other flavors a lot more since sweet was absent, and I could kind of start to detect the nuances between the other flavors in my food. Um, when I started to add sour to my food, like if I squeezed some lemon on a fillet of fish or something, it tasted sweeter for some reason, even though there's hardly any sugar in that at all. Um, I could start to taste the sweetness from starch if I ate something like a white potato. Um, some of the starch would break down on my tongue and it tasted a little sweet to me. Cucumbers, which, an entire cucumber has what, like 4 or 5 grams of sugar total. Those began to taste like watermelon. Well, not exactly like watermelon, but they're cousins, by the way, cucumbers and watermelon. But it doesn't have the sorbitol that watermelon has, which is a very sweet compound. But cucumbers, they started to taste like they were coated in sugar or something. So and again, this happened within a couple of days, not weeks, is all it took to kind of reset that set point. However, on the downside, I had unreal cravings for sugars. Just like I just craved sweets. And it wasn't too bad if I was out, running around doing stuff. But if at the end of my days when I didn't have anything to do or I was winding down, reading a book or something like that, that's when those cravings really started to hit me where it's like, I could really enjoy a bowl of ice cream right now or, whatever. So those are when the cravings would come through. And like in the workplace, if I would see somebody eating a jelly donut, that would kind of trigger those cravings too. I would look at it and be like, wow, that jelly donut looks really good. I would sure like to have a jelly donut as well. But I've always had a sweet tooth, and I think this protocol was actually very good for me to understand how my reward system had been hijacked and how I could get back on the right track. Unfortunately for me, throughout these 30 days, these cravings never went away. Even on the very last day, I couldn't wait for it to be over so that I could start just adding a little bit of sweetener to my coffee in the morning again or something like that. Although I did appreciate the natural taste of the coffee, and I'm from Louisiana, so I put a little bit of roasted chicory root in there too, and it tastes pretty good. But just having just a little bit of stevia in there in the mornings, just makes it so, just a lot more delightful. Another thing I did on this protocol, though, is that at some point, I cut out caffeine for 10 days because that jacks your dopamine reward system too, and I'll go over that in the next video. But so I wasn't even able to have coffee during those days at all. The other side effect that I got from this that was not good for me was I had an unreasonable amount of weight loss. And I was losing, I think anytime you're losing more than 2 pounds per week, there's an issue. And consuming more calories didn't seem to do anything as a result. I don't know if it was water weight or something, but I was shedding more weight than I had anticipated. I didn't actually anticipate weight loss at all as being a side effect of this. And I almost discontinued because I thought that it was putting my body in an unhealthy state. But I was able to make it through the 30 days, and I'm back on track now, so all of that's good. So be careful with this. If you start losing weight too fast, you may want to stop and cut it out. It's particularly if you're underweight. If you're overweight, you still got to be careful too. People who are overweight, though, can lose more weight at a time. It's not going to affect them as much. Depending on what your body fat percentage is, you could, if you're overweight, you can lose probably 2% of your total body weight per week. Whereas if you're within 15%, it's probably only 1%, and if you're lower than 15%, maybe 0.5% is kind of what you want to go with to be in a healthy range of weight loss. Anything over that can do metabolic damage. It can do a lot of pretty harmful things, and so you need to be careful. You never want to lose weight too quickly. So that was my experience cutting out sugar or not just sugar, but all sweeteners for 30 days. Do a reset. Now I don't need near as much sweetener in anything. I'm much more sensitive to it, and I'm going to try to keep these levels down. And if I feel like these levels are creeping up, I might do a shortened version of this later so that I can keep going back, just restoring my set point down to a normal level, because I think that's going to be healthier overall. And I think that the behaviors that are implemented by eating sugary foods or sweet foods or something, can be very damaging. So yeah, I give this protocol a shot. This is part of it. There's some other things too. And again, the goal is to kind of reset everything, recalibrate the brain, and get you back to baseline for all this stuff, because excessive amounts of caffeine, excessive amounts of looking at a computer screen, light, all this other kind of stuff can really hijack your brain, lead to an unhealthy lifestyle, create health side effects that you may not even be aware of. So look for part 2 of this next week when we talk about how to cut out caffeine from your diet and do a caffeine reset. All right, we'll see you next time. Like and subscribe, hit the notification bell, all that good stuff. Go to FixYourGut.com, read some articles, and check out our sponsors. All right, have a good one.